Welcome, everyone. I'm John Bagnulo with you this afternoon or this morning, depending on where you are in this country. Thanks for joining us for what I think is a very critical discussion or lecture around the potential for nutrition to improve outcomes for cancer patients, those who have either had chronic struggles with cancer or have recent diagnoses. I think it'll be hopefully very enlightening to those of you who have asked some of the questions that we've had over the past couple of years. As a company, we frequently hear questions regarding the importance of diet and or sugar carbohydrate restriction. And we think this will be really helpful to those of you who have frequently asked that question. Before I get started, I would like to note that everyone who is signed up for this, you will receive an email with a link to this recording this afternoon and to the presentation. You'll also receive a certificate with your the CEUs that you'll be given for participating in this lecture. So that will be coming in the form of an email shortly after this. I also want to thank Robin Gentry McGee for sponsoring this work and for her overall efforts to make this world a healthier place, for all that she's done with functional formularies. And I want to thank Brian McGee for his help with the slides and the artwork that I'll be showing you today. And I want to thank Catherine DeGeronimo for helping me with this presentation and all of the technical aspects of it. And I'd also like to thank, in advance of the talk, Mark Vincent, whose work on the press pulse model of biology and biological systems has really illuminated my understanding, and certainly many others who have a greater understanding of it than I do, and whose research careers have been centered around it, on the ability of a metabolic cancer intervention to improve outcomes. And in addition to Mark Vincent, I'd want to thank Thomas Seyfried of Boston College, as well as Dominic D'Agostino and Angela Poff of the University of South Florida, all of whom have shown just really incredible results in research with their therapeutic use of a ketogenic diet. So with that being said, as I mentioned earlier, we have frequently over the past several years, and I did before coming on board with functional formularies, regularly get asked the question, do I think that nutrition makes a difference with respect to cancer outcomes, or for that matter, the prevention of cancer? And I believe very strongly that it does. I want to share some of the most important points with you this afternoon that would support this. It's just, we're at a point now where if you look at the last several years, from 2014, for instance, to 2017, deaths from cancer have risen around 3.45% or so, and new cases incidence of cancer has risen about 1.7 to 1.8%. So we've had an increase in both the incidence and even more so the death rates from cancer. We know that nutrition really, is something that is compromised in most populations here in North America, and that's becoming a global pandemic as well. So the two are linked closely, and I really hope to explain some of the differences in terms of common perceptions of what cancer is and what more recent research has shown to be true. Now, most of you have probably heard the parable of different people who've had whether it be blindness or other impaired senses, trying to describe the same thing. And most of us have heard about the efforts to describe an elephant when each person is holding a different part of it, and that's certainly the case with cancer. I think if you take a look at most people look at cancer as being this genetic defect type of occurrence where people have exposures or are dealt bad genes, and this causes problems with different aspects of their cellular physiology, most notably the DNA, and then you get uncontrolled cellular division. And what I'm presenting today, which of course is not my, it's not my research. It's those of many others, but it certainly relates closely to the type of nutrition that I practice and that I recommend for people as an educator, is that this notion of cancer being a random genetic event, should really be challenged, and we should look at cancer as more of a metabolic disease. And the reason being is, here's a slide that, for instance, shows a more contemporary perspective on cancer being the result of just a gene defect or mutation, it's more contemporary because inflammation is actually a contributing cause to this, which we know is certainly true. Inflammation is closely related to cancer. But what I'm proposing today, for a better understanding of the role in carbohydrate restriction and the use of certain plants, supplements for that matter, which we'll talk about towards the end of this, is that it isn't strictly about a genetic defect causing everything downstream from it. It's more about the mitochondria, and that's why I showed an image of the mitochondria a few slides prior to this. All cancer cells have some very, very similar aspects. While they may be very heterogeneous in some ways, almost all cancer cells have a very, very significantly impaired or limited, some have no capacity at all for mitochondrial respiration. When I'm talking about this, I'm talking about the full use of the Krebs cycle to produce energy. And that is, of course, if we think about aerobic respiration, it is the primary source of fuel for most of us in a healthy state and for most mammalian cells. And that's where you have 2 carbon units in the form of acetyl-CoA being run through this citric acid cycle or Krebs cycle, producing 32 to 34 ATP molecules. And I know many people probably cringe when they think about the Krebs cycle and what they had to memorize in a biology course or an anatomy and physiology course somewhere along the way in their education. It is a fundamental component of understanding why carbohydrate restriction really improves outcomes when it comes to cancer intervention. And again, this is, in many ways, this is a very, very important concept when you understand human evolution and you take a look across all biological systems. And this is where Mark Vincent's press pulse theory is so applicable to treating cancer cells, because cancer cells in many ways are like a species unto themselves. And all species really change when they have a press or chronic stress coupled with a pulse or more acute stress. That's what the press pulse theory means. You have chronic stress coupled with a pulse or acute stress, and this leads to the extinction of species. And almost all species have gone extinct when they've had both a chronic and acute stress placed upon them at the same time. And that's what we're talking about. We're talking about treating cancer cells as though they're their own species, which they are, because they don't have metabolic flexibility. They don't have the ability to burn all fuel sources like normal healthy cells do. And that's really, in summary, what I want to explain and why I think it's critical that people reduce the amount of sugar in their diet, not only for cancer intervention to improve outcomes as part of a cancer patient's treatment, but also for preventative reasons. Dietary sugar, hyperglycemia, which occurs with that, hyperinsulinemia, these are all massive risk factors for cancer and do not have favorable outcomes associated with them when they coincide with cancer. So the press pulse theory explains that cancer cells survive and have been looked at as being very difficult to eradicate because they have a very unique ability to rely not on aerobic respiration, but to rely on glycolysis, which is where sugar generates a small amount of ATP, but with an abundance of sugar, cancer cells will rely on this even though their mitochondria is not functioning. And along the way, they also produce lactate, which is certainly noteworthy in this conversation. If you look at the bottom of this slide, you'll see that as lactate production in the orange line increases, and oxidative phosphorylation, which is what we talk about, being required in the mitochondria as part of the Krebs cycle, as that goes into decline, that's when you get the most rapid advance or progression in cancer. But this is really the genetic defects that we find in cancer cells are going to be more downstream from damage to the mitochondria than they are going to be the initial cause. And I think that's what's really important. And it's not new. Otto Warburg, made these observations, 80 or 90 years ago And, you know, it's never been disproved. The Warburg effect, as it's known, is where all cancer cells have impaired ability to generate energy with respiration and have to rely on glycolysis and the fermentation, which just means producing lactic acid from glucose. They have to rely upon that for their survival. They can also use certain amino acids with their impaired mitochondrial function. They can use certain amino acids to produce modest amounts of ATP as well as nucleotides for generating new cells. Glutamine is a noteworthy amino acid that is used by cancer cells. And Warburg observed this, again, decades ago, and it's never been disproved, and it's really now serving as a foundation for the research of those that I mentioned at the beginning of this talk in various areas around the country, where whether it's in an animal study or it's a case study, those that are placed on therapeutic ketogenic diets are seeing, remarkable turnarounds in their cancer. So normal cells, as I mentioned earlier, will take glucose and will generate a small amount of ATP or energy as that is run through glycolysis initially, and then it will enter the TCA or Krebs cycle, which takes place in the mitochondria, and this will produce, large amounts of ATP, which, of course, is not strictly going to be dependent on glucose. Fats are going to provide a significant amount. Ketone bodies can enter the Krebs cycle. Certain, skeletons from amino acids as well can enter the Krebs cycle and can give us significant quantities of ATP. But in a cancer cell, you have no ability for this aerobic respiration, and everything comes down to glycolysis and or the use of things like, as I mentioned earlier, glutamine, which will use single enzymes from the Krebs cycle to produce smaller amounts of ATP. But because there's usually an abundance of both glutamine and glucose available in circulation, most cancer cells have no bottleneck in terms of their energy supply. So even though they may have impaired aerobic respiration, they're able to divide uncontrollably and grow very rapidly because there's just so much glucose and so much glutamine and other amino acids available in circulation. But glutamine is certainly the most noteworthy of all these amino acids that have the potential to feed cancer growth. And when this happens, there are, again, certain enzymes that become upregulated. One of the most noteworthy that you'll see towards the lower left-hand corner of this slide is pyruvate dehydrogenase. Now, pyruvate dehydrogenase is something that is going to really fuel the conversion of this over to lactate. And lactate is a critical byproduct of cancer cell metabolism of glucose because it has many other effects on normal check and balance systems that would stop cancer cells from dividing or continuing to divide uncontrollably. And one of those processes would be apoptosis. Apoptosis, of course, is cell programmed death. And we know that apoptosis is suppressed with higher levels of lactic acid. We also know that angiogenesis is fueled or stimulated with higher levels of lactic acid. This is one reason why reaching an anaerobic threshold as an athlete can fuel angiogenesis in muscles that are being trained. And that's a good thing in exercise physiology, but it's not a good thing when that angiogenesis is taking place in a tumor because the cancer cells are producing more lactic acid. So it's really about the application here. So these enzymes in red are upregulated. You can see hexokinase, phosphofructokinase, all things which really feed glucose metabolism. Now, again, I mentioned the p53 gene. That is the gene that, when expressed, which normally would happen if you had impaired mitochondrial respiration in an unhealthy cell, you would expect to have the p53 gene expressed. Apoptosis would soon occur, and that cell would be eliminated from the cell pool within that tissue. But this does not happen. This normal end of cell life with apoptosis does not occur because the higher levels of lactate suppress that gene's expression. This is not only true of cancer cells, but also with things like polycystic kidney disease. This has been shown with other polycystic ovarian syndrome or disease. A lot of higher rates of neoplasm are closely linked to this Warburg effect or total reliance on glycolysis by cells. And again, going back to, you know, some of the other important aspects of an impaired glucose, impaired aerobic respiration, or the increased reliance of cancer cells on glucose, glutamine is a big, you know, a big part of this, and that's why, as we get into what a more therapeutic diet looks like, this is why it may be beneficial to restrict protein intake to levels of somewhere, you know, between, you know, 15 and 10% of total caloric intake, because larger quantities of protein may increase the glutamine pool to the point where it would feed cancer cell growth as well. So, you know, again, there are many wrinkles to this metabolic approach, and it isn't solely about carbohydrate restriction and, of course, reducing available sugar, but it's also about keeping protein intakes lower than many Americans would consume. And if we think about many of the pathways that would also be favorably affected with glucose levels that are lower, less available glucose to cancer cells, more ketone bodies, you know, you can look at things like insulin-like growth factor, you can look at things like the mTOR pathway, PI3K. All of these are pathways that we know play pivotal roles in the growth and development of cancer. And many people are told that they have an abnormal pathway in one of these, and that their drug they're being given as part of a chemotherapy you know, protocol may target that drug, and that may be effective. But I think it's very important for us to understand that restricting sugar, restricting carbohydrates overall, keeping protein intake under a safer percentage of total calories in a person's diet, all of these things would have very favorable effects on these pathways as well, especially things like IGF-1 and mTOR pathways and the cancers that they will drive. So this is, you know, in a way, it's a kind of a shotgun approach to having very favorable effects on cancer metabolism, because you are starving cancer cells of their primary fuel. They're not metabolically flexible like our healthy cells are. And while we do this, we also shut down a lot of the pathways that we know have been identified as being primary driving forces behind cancer cell growth. Now, when we look at, again, diabetes, most people have an understanding that, you know, diabetes is a function of insulin resistance, and that, you know, as insulin starts to occur at higher and higher levels at any given time in a person's bloodstream, we tend to store more of our calories as fat. We tend to have a greater risk for cardiovascular disease, for high blood pressure, for all of these, you know, components of the metabolic syndrome that we know is, again, it's an epidemic. What many people fail to appreciate is that all of those same biological patterns that occur with hyperglycemia and hyperinsulinemia also feed cancer and feed cancer cells. Again, a lot of the pathways that I just mentioned with the previous slide, insulin-like growth factor, mTOR, PI3K, all of these are also stimulated by higher levels of insulin. So you have a double-edged sword. You have higher levels of insulin, which directly stimulate pathways that we know feed and promote cancer cell growth, tumor growth, angiogenesis. You also have higher levels of sugar or glucose in the bloodstream at any given time, greater amounts of glucose available to cancer cells, which can be used for higher levels of metabolic consumption. So again, lowering insulin, lowering blood glucose levels, keeping sugar at lower levels, while we also, as we'll talk about in a moment, try to raise circulating levels of ketones, which have a wide variety of beneficial effects. I mean, these are axiomatically beneficial effects and should be recommended for patients who have cancer and may be on one or more chemotherapy regimens, radiation. Regardless of that, people should be taught the importance of lower amounts of sugar, carbohydrate restriction. Even if it doesn't approach a ketogenic diet, it makes sense that we don't add more sugar to an already burning fire within the body. Unfortunately, it's not talked about this way, and people are told to eat whatever they want while they're in these various regimens of conventional cancer treatment. And it just makes very little sense once we understand metabolically what cancer needs and what fuels its growth. So again, whether you're talking about the effects of the excess sugar in the bloodstream, whether you're talking about the effects of insulin on processes such as angiogenesis, we also know that IGF-1 increases, IGF-1 binding protein decreases. That's another risk factor for whether it's prostate tumor, breast cancer. All your cancer types really are, negatively affected when it comes to IGF-1 to lower levels of IGF binding protein. So there's numerous reasons why we want to get control over glucose control in the body and the amount of available glucose. When we, take a look at this, we know that, again, insulin resistance, and obesity go hand in hand, along with other aspects of the metabolic syndrome. So people who are obese, who have metabolic syndrome, they are at a higher risk for cancer, for most types of cancer, breast cancer being most noteworthy, colon cancer being also very high on that list of associated cancers with obesity. And we know that, interestingly enough, the use of metformin as a diabetes medication, blood glucose control medication, has had very favorable effects in limited numbers of trials for cancer patients. The reason being is that metformin lowers glucose levels. It reduces available glucose and that which would be available to cancer cells. It also has very favorable effects on activated protein kinase, AMPK, and that would be a primary example of a pulse or acute stress that we want to place on cancer cells, whether it's metformin or using some type of plant compound that we can discuss a little later on, or a very traditional chemotherapy. Those are pulse stresses or acute stresses that would add extra stress to cancer cells that may have a more chronic stress placed upon them with a reduced availability to glucose and or protein. So it's interesting that diabetes patients often are given metformin, and metformin has been shown in clinical trials to have benefits to cancer patients. So it's a prime example of how glucose feeds cancer. Drugs that reduce circulating glucose and have other favorable effects, such as those on pathways that feed cancer growth, would be a prime example of a pulse intervention. Now, on the topic of ketogenic diets, I often hear that people are interested in this, whether that be an oncologist or another primary care physician that may be looking over the health of a patient with cancer who's also seeing an oncologist, family members, and then patients themselves very interested in ketogenic diets. What are the potential benefits? And I think that we're learning basically every month. If you go on PubMed, you start looking at peer-reviewed scientific journals that are primarily looking at animal studies, but more and more are looking at limited numbers of human trials, that ketogenic diets hold great potential for slowing or stopping cancer growth because of these metabolic bottlenecks, so to speak, that are placed on cancers and on tumors. And when you start to look at the strength of those, it's now spilling out into many other areas, not just into cancer, but into neurological conditions, neurodegenerative diseases, and a variety of others. So there's a growing body of evidence that the ketogenic diet and carbohydrate restriction, coupled with at least modest protein restriction, is the way of the future in co-treating cancers with other, whether it be drugs or other potential compounds. Now, the primary arguments against a ketogenic diet and against significant alterations in the amount of carbohydrate or protein in a patient's diet is that it's going to have associated health risks to the patient, or that it's going to be too costly to the patient. I find this incredibly puzzling, to say the least, given the fact that if you take a look at some of the most conservative estimates, such as those that have been published in a variety of places like News and Observer and Time Magazine, there's countless assessments of here what drugs cost. When you take a look at something like Avast and you look at what it costs per dose or per month, and you start running the numbers on these things. I mean, no ketogenic diet. I don't care if someone's eating in a 5-star restaurant every night and they're eating ketogenic at an institution like that or a dining facility like that. It's not going to approach the cost of these drugs. I think that's a ridiculous argument, cost, especially when you take a look at the cost of drugs that are being used. And then when it comes to the safety of a ketogenic diet, I want everyone to understand we're not talking about ketoacidosis where you have millimole levels of 12 or 13. We're talking about millimoles of maybe 4 or 5. And even there's some great benefit to levels in the 2s, 2 to 3. So this is not a starvation or acidosis type intervention here. We're talking about modest levels of ketones in comparison to those types of extremes. In addition to this, some people would suggest that a ketogenic diet, and there's some again, there are a lot of misconceptions around what that is. I think people hear ketones and they start thinking starvation or Atkins type diets, and that's not what this is about. This is really very different. And the research from a cardiovascular investigation, type of cardiovascular trial perspective is that it's very favorable when you start to take a look at the small dense LDLs and triglyceride levels, and you start looking at HDL levels and all of those things that we know are more qualitatively reflective of cardiovascular health. The ketogenic diet is also very heart healthy. There really aren't any risks associated with a ketogenic diet when it's done properly. So, I just find that those arguments against a ketogenic diet are interesting given the cost of drugs and the research supporting it in other areas of health. Now, what a ketogenic diet actually looks like can vary from researcher to researcher and from how well it is going to work from an individual to an individual. Young children may be able to tolerate a ketogenic diet that looks like this, where 20% of the calories come from protein and 75% of calories come from healthy fats. And that might work well for an adolescent who have more metabolic requirements for growth and development than, let's say, a 65 or 70-year-old individual with cancer. They may need to have protein restricted to 8 to 10% of calories. But in general, a ketogenic diet is going to be somewhere around 75 to 80% of calories coming from fat, somewhere between 8 and 20% of calories coming from protein, and anywhere from 5 to 10% of calories coming from carbohydrates. Now, the quality of all these, I think, is very, very important. I think we should emphasize healthier fats that are predominantly saturated and monounsaturated, not a significant quantity coming from polyunsaturated seed oils and things like that. I think having some flaxseed oil may be of benefit, relying heavily on olive oil, things like macadamia nuts, coconut, avocados, of course. These are all wonderful fats to rely heavily upon. But olive oil could be certainly a staple in the diet of anybody on a ketogenic diet. Protein should come from a wide variety of sources, in my opinion, and I think that it can come from grass-fed beef. It can come from things like lamb, bison, but it can also come from a wide variety of fish, poultry, eggs, and certainly dairy. Higher fat dairy products can, while they do contain carbohydrates in the way of lactose sugars, it's going to be well tolerated as long as they're kept in moderation. And then carbohydrates should be emphasized from root vegetables, things like sweet potatoes, carrots, beets, parsnips, but certainly people can get a small amount from berries and other fruits. This may sound very radical or extreme, but we have to think about the condition. We have to think about cancer and what's going to be required to put a metabolic pressure on those cancer cells. We can't have everything in moderation. I know that's what a lot of people are taught as part of their, registered dietitian curriculum, that they learn everything in moderation does not work with cancer. The cancer cells are too good at finding carbohydrates in a wide variety of foods. So cancer cells really have to have this kind of pressure placed on them. And if people are having, significant quantities of dietary sugar from things like large amounts of fruit juice, excessive grain, flour products, when they start to, represent a significant percentage of calories, the cancer cells are going to find the sugar there. You're not going to be generating ketones, which are an interesting, byproduct of this carbohydrate and protein restriction. So it's really important that numbers are somewhere between 5 and 10% of calories coming from carbohydrate. Again, children typically do much better with, or can tolerate slightly higher amounts of both protein and carbohydrate. And these meals can, again, be very healthy. They can still be plant-based. You know, salads, I think, are a great thing to rely on for maybe one of a patient's meals per day, but it doesn't have to be all about salads. You know, olive oil, I mentioned, is a great staple, but you certainly have a wide variety of fats and oils that could be sourced from things like coconut oil, grass-fed butter, even conventional butter, I think, can work really well for people on a ketogenic diet. Heavy cream, there's a wide variety of choices. And then you can have, as I mentioned, beef and vegetables, salmon with guacamole on top of vegetables. A lot of meals will work as part of an overall ketogenic diet. So that would be an example of metabolic restrictions in terms of reducing carbohydrate availability to cancer cells, a chronic stress where we're not allowing those cancer cells to rely on that glycolysis for growth and development, or the fermentation of glucose into lactate. Then additional methods of pressure, which may be still chronic or could be more acute as a pulse application, would be the use of particular plant compounds. These are widely available. There's significant research supporting their use in both human and animal trials. And I have all of these referenced, a very lengthy list of peer-reviewed articles that are at the conclusion of this talk or at the back of this presentation, where you can look at the research that supports the use of three compounds, which I feel have maybe the greatest overall scientific evidence supporting their use. These are Nigella sativa, often referred to as black cumin seed oil, curcumin, which is the leading bioactive compound in turmeric, and then green tea compounds, e.g. CG, epigallocatechin, which is, you know, again, it's the most medicinal compound that we've looked at yet in green tea. So these are things that a lot of people can use in combination with a conventional, cancer treatment. I am not suggesting that these are cures on their own, although some of the research, may suggest that. I'm suggesting that these should be used concomitantly, looked at closely by an individual with cancer and their oncologist to see if it's a good fit for them. But again, the peer-reviewed research on these is very clear that there's a potential for them to work for many forms of cancer as a co-treatment to improve the outcome. And these can often be placed on top of, again, a conventional treatment, as well as something like a ketogenic diet. And I feel the more of these pressures that we can apply on cancer cells, the better. I'll start with Nigella sativa because it's one that many people have never heard of. Again, often referred to as black seed oil or black cumin oil. Its primary compound is thymoquinone, and thymoquinone is a very interesting compound with a benzene ring at the core, which a lot of cancer drugs have been developed around. Thymoquinone is found in a variety of other plants in addition to black seed oil. It is in cumin, it's in some other herbs and spices, but black seed oil is the richest source of it. And we start to look at all the different ways that thymoquinone, where research has shown it to have positive influences on one or more aspects of cancer metabolism, growth, or on processes within the cell that would bring an end to that cancer cell's life. Again, apoptosis being a primary example of that, but there are others as well. There are just multiple ways that thymoquinone, again, coming from black seed oil or Nigella sativa, can favorably affect cancer cell growth or limiting its growth. So when we take a look at the research supporting this, what most of them show is that it inhibits, from a preventative standpoint, it inhibits carcinogenesis. It can be very protective to the mitochondria and can really prevent a loss of this mitochondrial respiration, which is fundamental to this talk this afternoon. It can also induce apoptosis. It can also inhibit angiogenesis. So it has, again, all of these abilities to influence or intervene within cancer growth from an initial point all the way to those which would help prevent the growth of blood vessels into a tumor that has already become established. And So many drugs have been developed around substances like thymoquinone or sulforaphane, which is found in your brassicas, EGCG, which we'll talk about in a moment. You take a look at all of these, chemo drugs, and many of them are based on plant molecules, which have very similar effects. And again, I always emphasize that I'm never, whether we're talking about, chronic diseases that are in the area of cardiovascular disease or glycemic control, I don't think we need to turn our back on Western medicine or on the use of particular drugs, conventional treatments. I just think it's important to appreciate all potential interventions, whether that be, conventional drugs, chemotherapy agents, radiation, of course, surgery. But we should consider many other interventions to have benefit and that can be used mutually, if not, exclusively. So I think it's just important to appreciate these as very viable options that patients and the families of patients have to look at more closely and to discuss. The dose of thymoquinone on, for instance, the p53 gene expression and on cell viability is really clear. More thymoquinone from more black seed oil has very favorable effects on breast cancer cells not surviving, which is what we want. So it's one of those things where, greater exposure to thymoquinone through multiple dosings at each meal produces more favorable benefits. And this has been shown in vitro as well as in vivo. Now we'll talk briefly around turmeric, which is, again, the richest source of curcumin that we know of in any spice or plant compound. It's widely used in Middle Eastern and in Asian, Indian cuisine. It's a very interesting compound because it's not very bioavailable when it's just taken alone or is eaten in raw form. Curcumin really requires cooking for it to be more bioavailable. It should also accompany a high-fat diet, and it may require the use of something like black pepper or bioperine to improve its bioavailability across the brush border of the intestinal wall. It's very, very, very interesting when you start to take a look at curcumin. It's very similar to what we showed with respect to thymoquinone previously, that it has so many favorable effects on human physiology and on our biochemistry. It's just really amazing when you take a look at the potential for this substance biologically to improve outcomes, not just within the area of cancer. But if we do focus on cancer, most of the biomarkers that we look at in animal models of cancer improve when animals are given curcumin. Now, of course, the dosage is often quite high. It is a blood thinner, so that's really an important part of a conversation you'd have with your primary care physician or with your oncologist when you would be recommending curcumin at a therapeutic dose for someone with cancer. You'd want to make sure that they were taking a look at their INR, their prothrombin time, or some way of, you'd have to be assessing their clotting time, because it is a very significant anti-clotting agent, especially when we're talking about using it in the dosages that would be required for therapeutic cancer intervention. But when you do take a look at the research around its ability to have favorable effects on cancer biochemistry or cancer metabolism, it's a significant mTOR inhibitor. It tends to have very, very favorable effects, as we mentioned earlier, on things like apoptosis and autophagy, which is the process of recycling damaged cellular components, and that's really important when it comes to most lines of cancer cells. So curcumin is, again, something that's not very expensive, needs to be cooked with. If it's going to be part of a menu, if it's going to be basically woven into the recipes that a cancer patient would go home with, you're talking about using anywhere from 2 to 3 teaspoons to receive the amount of curcumin that most animal trials have shown to have favorable effects. 2 to 3 teaspoons per meal, 2 to 3 times per day. That would require at least 5 to 7 minutes of simmering in either water or a monounsaturated or a saturated fat-based oil. That can be part of a stir fry. That could also be part of a tea or something like that. So it requires heat and maybe the use of something like black pepper to further enhance bioavailability. Lastly, we'll talk about green tea and about maybe the use of green tea extract. Green tea is something, of course, that has been a big part of Asian and in particular Japanese culture for thousands of years. The primary component to green tea and to green tea extract is EGCG, a form of catechin, epigallocatechin, which again inhibits angiogenesis, favors apoptosis, has favorable effects on things such as AMPK, activated protein kinase, and can imply, again, a very acute type of stress to cancer cells, or a more of a pulse stress that we've been talking about this afternoon, that can limit one or more biochemical pathways critical to cancer cell growth and development. You can take a look at, and when you look at this slide, at the amount of EGCG in terms of micromoles that were required in animal studies to produce favorable effects in these various areas. And some of these levels of micromoles are very, very high. When you take a look at MAP kinases, for instance, some of the research required levels of 20 micromoles. That's a lot. You know, that's something that, you know, would only be acquired through the use of an EGCG supplement. You would not be able to, you know, physically drink that much green tea. However, much lower levels were shown with things like angiogenesis, where levels were only 0.5 micromoles. That could easily be acquired by drinking green tea a couple times during the day, as long as it was a high quality green tea and actually contained the EGCG, something like a matcha green tea or a sencha green tea that was steeped at around 180 to 190 degrees. That would easily be effective to help a patient get to levels of 0.5 micromoles. So the levels vary. Quite often, many of these compounds are dismissed by skeptics of plant compounds in improving cancer outcomes, and they say that the amounts required are not achievable through diet or supplement use alone, and that's just not the case. Many studies have supported physiological levels that are attainable through both diet and or supplement use. Again, EGCG has numerous interactions with surface proteins and channels on cancer cell walls or membranes, and this is a big part of reducing certain cancer cells' ability to metabolize fuel sources or to acquire higher levels of those hormones, which may be used to drive higher levels of growth. So there's multiple interactions taking place between this compound in green tea. I have included what I consider to be a landmark paper. This is the whole press pulse strategy for improving cancer outcomes and the management of cancer. Lead author is Thomas Seyfried. You can look at Thomas's talks. He has several YouTube talks that are available. He's given a wide variety of talks around the country to physician groups and to those who deal with cancer patients. And I really find his work and his talks very inspirational. I also want to make note of his and Dominic D'Agostino's development of the blood glucose ketone index. It's a very important tool for using and monitoring a cancer patient's progression in a ketogenic diet. It's a very simple formula that he and Dominic D'Agostino and others such as Angela Poff have used to look at the effectiveness of a ketogenic diet in humans as well as in animals. It's very simple. You have to get blood glucose levels measured with a glucometer, and you convert that to millimoles, which is simply divide by 18. That gives someone a very simple blood glucose reading in millimoles. Then you divide that by their ketones in millimoles. And they've shown that your blood glucose ketone index, again, glucose and ketone levels have to be taken at the same time. If you use that index, it can clearly show you whether or not your patient is in the target or therapeutic range for restricting cancer cells of glucose, as well as supplying the body with an available amount of ketones, which have so many favorable effects on healthy cell and as well as cancer cell metabolism. So again, Thomas Seyfried, Dominic D'Agostino, you can take a look at this paper. You can look at their blood glucose ketone index and recommend that for those that you work with. And I mentioned that I had references here. I have them for the ketogenic intervention to cancer. I have those for the use of Nigella sativa or black seed oil. There's actually more with Nigella sativa than almost any plant compound we could look at, including garlic, when it comes to to cancer and other chronic diseases. So that list is very extensive. And then curcumin and green tea, I tried to choose those within the last 2 or 3 years because there's a large amount of references for all of these. And with that, I really want to thank you. I hope that this has in some way spurred your curiosity and will help you delve deeper into the use of metabolic therapy to improve the outcomes of those that you work with. Again, maybe not in place of conventional treatment, but used concomitantly to greater improve the likelihood of a patient's response to more conventional treatments. Thanks so much, and I look forward to talking to you again sometime.
John, this is Catherine.
Yeah.
Thank you so much for a wonderful presentation. Since we have some time, there were several questions. Do you mind looking in the question box and commenting on some of them?
Sure. I'm trying to bring up the question box and can't seem to do that.
I can read them too if you'd like, if that's a bit easier. So
if you, yeah, that would be great.
Okay, sure. The first, if people are using high doses of glutamine to try to heal intestinal permeability, are they increasing their risk of cancer?
That's a great question. I don't know the answer to that, but I know that I would not recommend that anybody use glutamine who has a cancer diagnosis or knows that they have cancer. I would not recommend glutamine therapy for those individuals, but I'm not sure if the amount of glutamine being used to try to repair intestinal wall function is going to be a risk factor for cancer. I have heard this asked before. I will say that glutamine is found in a wide variety of protein sources. It is the amino acid that we have very, very large amounts of typically in circulation. So, you know, it's most of us have large amounts of glutamine to begin with in circulation who are eating any type of or significant amount of dietary protein. I'm just not sure if taking larger amounts as part of a glutamine therapy, if it pushes the amount of glutamine up in circulation, you know, to the point where it's a risk factor. But I would not recommend it if someone has cancer. And I know that some people, unfortunately, have used glutamine therapy while they have cancer. I would not recommend that.
Okay. How do you realistically see a high-fat diet be consistently consumed by a patient who suffers from anorexia and nausea? If you are cutting carbs and protein and driving a high-fat diet needed to promote ketogenesis, where is the patient getting any calories at all?
Well, they will be receiving adequate calories. That's not the issue. I think that the first part of that question, which would be, you know, having nausea, I think that's a much more, you know, valid question. I think that, you know, the ketogenic diet would provide adequate calories. Of course, it has to have ample amounts of fat, and I think the fats have to often be blended with foods to have greater palatability. It requires some creativity, either on behalf of the registered dietitian that's working with the patient, or on the primary care physician if they're willing to embark on this, or on the patient themselves or the family member. I think that blending healthy fats such as avocados into things like guacamole, using fresh squeezed lemon or lime juice, which have virtually no significant carbohydrate content, using herbs and spices, and being really creative with the way foods are cooked. You know, stir fries can be amazingly palatable, very, you know, just delicious meals overall. So we're not talking about a ketogenic diet where people are, you know, just swallowing tablespoons of butter or coconut oil. That's a very common misconception, I think, that's portrayed by people who are very skeptical or critical of a ketogenic diet. They don't think that it's possible. It's very possible. You know, thousands of people are doing this every day for one or more reasons, not always cancer. There are a variety of books out there, you know, written by people like Angela Poff and others. Dominic D'Agostino has one. There's just great books out there that show you how you can do this. But I think it's very feasible, and I think that you know, people who suffer from nausea are maybe more challenged, but it's just going to require that they refine or fine-tune the types of fats and oils they use and the way that they are combined with things, either in a blender or in a recipe that requires some cooking. But getting enough calories, that's not an issue.
Okay, so this is a two-part question. There is research that shows poor outcomes from weight loss during treatment. Can you talk about this in regards to a ketogenic diet? And the second part is, can you discuss fat intake and inflammation?
Yeah, sure. I think that the poor outcomes associated with weight loss are really difficult for people to pull all the variables apart. If you have someone who has a rapidly progressing cancer, they become, basically cachexic because the cancer's metabolism is so great that it's robbing their body of everything it needs. And I think that, you take a large number of studies that, pool all of these different individuals together at various stages of cancer, you're going to find that people with cachexia, of course, they have a much poorer outcome than those who aren't at that point yet. And you're going to see an association between weight loss and poor outcomes. Absolutely. What we're talking about, though, we're talking about limiting cancer cells' metabolism of carbohydrate and protein to a lesser extent. It may produce a certain amount of weight loss. Most people who do go on a ketogenic diet do experience some type of weight loss, but I think it's really important to understand that you are limiting the cancer cells' available primary fuel sources, and that's going to maybe produce some weight loss because as insulin levels drop, as the amount of IGF-1 drops, and all these things are going to produce some weight loss in an individual. But most importantly, you are starving cancer cells of what they need to grow. And this is really important that we separate this from the type of weight loss that occurs because of cachexia. Unfortunately, it's all lumped together. And people say, well, if you go on a ketogenic diet, you're going to lose weight. And if you lose weight, you're going to have a poor outcome with respect to your cancer. You can't really blend these two things together. We're talking about weight loss that may occur because someone goes on a ketogenic diet. Even though they get enough calories, their insulin levels are going to be lower and they're going to lose some weight. But that's very different than the type of weight loss that occurs when people are eating, large amounts of calories. They're getting lots of carbohydrate and they're still losing weight. That's because of cachexia. So, these two things can't be blended together, but unfortunately they are. And that's, again, something that is put out there as a counter argument against any type of dietary restriction. I so often hear people come from their oncology visit and they're told, eat whatever you want. Whatever we do, we want to make sure you keep your weight on or you gain weight during this cancer treatment. You don't want to lose weight because weight loss is associated with poorer outcomes. Again, this is, blending everything together. And I think people have to come out of that first visit with their oncologist or their physician or their registered dietitian. They have to be told, look, you can eat much healthier than a standard American diet when you have cancer. You can limit your sugar intake. You can maybe restrict your carbohydrate intake, even if you don't go into a full-blown ketogenic diet. And if you lose a little bit of weight because your insulin levels are coming down, let's not look at that as a risk factor for a poorer outcome with your cancer. Was that the first and second part of the question, or was that just the first part?
The second part of the question was, can you discuss fat intake and inflammation?
Yeah, sure. I think the fat should predominantly come from saturated and monounsaturated fat sources. We need to be very careful around especially omega-6 fatty acid rich seed oils. Those would be things like canola oil, sunflower oil, safflower oil, of course corn oil, all your vegetable oil blends. These are very, very high in their omega-6 fatty acid content, very low in their omega-3 fatty acid content, and that ratio is important with respect to inflammation. So that's when I would place the most important or primary focus on when it comes to fat choices. Try to keep fats saturated and monounsaturated. I have very little preference for either one. Both are very healthy for the mitochondria. Both are going to be more stable, and they're not going to produce swings in that icosanoid or icosanoid hormones such as prostaglandins, thromboxanes, leukotrienes. Those can be heavily influenced by too many of your seed oils. So I do think it's very important that olive oil, avocados, coconut oil, macadamia nuts, butter, heavy whipping cream. I think those would be the best fats and oils to use.
If most of the antioxidants exist in fruits and vegetables, I don't feel comfortable about restricting this food group, especially when we don't know all the benefits of produce. Help me wrap my mind around this.
Sure, I'll help that person. I'm not talking about limiting vegetables. I'm talking about limiting fruit. Fruit contains a significant amount of bioactive compounds. I received my PhD looking at the benefits of blueberries and anthocyanins. So I fully appreciate the benefits of these plant compounds. And I'm not talking about restricting vegetables. People can eat all the vegetables that they want. They, of course, would have to restrict those vegetables that contain larger quantities of starch, i.e. sugar, things like potatoes, sweet potatoes even. You cannot eat these in copious quantities where your carbohydrate levels are going to rise too much within the diet. Fruit does require a more significant amount of restriction. I think people on a ketogenic diet can still eat berries, anywhere from 1 to 1.5 cups. Some people can even eat as much as 2 cups of these berries a day, because berries contain very, very low levels of sugar and have, you know, much larger quantities of some of these compounds that I think this participant is talking about. Lemons and limes, of course, are basically free foods. They contain insignificant amounts of sugar. Grapefruits are well tolerated by many people on a ketogenic diet. So this is not an across-the-board restriction on fruits and vegetables, certainly no restriction on your non-starchy vegetables, and a modest restriction on maybe certain fruits. So, you know, again, I don't think this is, you know, this isn't a all or nothing here when it comes to fruits, and it's certainly not a restriction on non-starchy vegetables. So the two they're not mutually exclusive. Hope that helps.
Okay, so we might have time for one more question, and perhaps since there's so many, we can do a follow-up webinar in the future. But this is another 2-part question. Have you ever worked closely with a patient going through chemotherapy? And the second is, how do you suggest patients maintain significant fiber each day with restriction to whole grain carbohydrates and fruits?
Yes, I've worked with many patients who have been going through chemotherapy, including my son, and again, many others over the last 20 years that I've worked as a registered dietitian. And when it comes to fiber, I have to reiterate this. This is very similar to the last question. We are not talking about restricting vegetable intake. People can have zucchini, summer squash, cucumbers, radishes, you know, even, you know, of course, all the salad greens, your non-starchy, dark green leafy vegetables like collard greens, broccoli, cauliflower. People can eat fermented vegetable dishes like kimchi or sauerkraut. There's just such a wide variety of fiber-rich vegetables that, you know, whole grains they're not the gold standard for fiber to begin with. And it's unfortunate that if we think when we cut out grain, that fiber is somehow going to disappear. So vegetables are really what we should get most of our fiber from. When you look at the research, it is the type of fiber that's protective against a wide variety of diseases, cancer included. That's not always shown with the fiber that comes from grain. So I don't look at this as there being any downside to eliminating whole grains. I think that those can easily be replaced with high fiber vegetables, and I think that's really the way to go.
Okay, and John, how many more questions do you have time for?
You know, I have time for probably 2 more.
Okay, let's see. Is the high-fat diet recommended for only a short time? Does the high-fat diet increase the risk of diabetes?
No, I don't think it's only recommended for a short time. I think it's a function of a variety of variables and factors, such as the patient's age and how they're progressing. I think that people can, again, they can come on and off a ketogenic diet depending on how they've responded. And I don't think it's a risk factor for diabetes. I think you can take a look at a wide variety of published papers that show incredible benefits to higher fat, lower carbohydrate diets. And those may not all be ketogenic. Some of those, may show that, carbohydrates restricted to somewhere between 20 and 30% of calories are immensely beneficial for blood glucose control and reversing diabetes. So again, we're not simply talking about cancer here when we talk about a higher fat, lower carbohydrate diet. And I think there's a lot of confusion around what's a high fat diet. A high fat diet is one that's, typically 50% of calories coming from fat. That's not ketogenic. Ketogenic is when you get into the 75% of calories coming from fat. So I don't want to create confusion here between a high fat and a ketogenic diet. And what I'm really talking about is this afternoon is about the use of carbohydrate restriction and or ketogenic diets to reduce the amount of available fuel for cancer cells. But that being said, I do not think that a high fat and or ketogenic diet increases a person's risk of diabetes. And I think that people can be on these for very, very long periods of time without any observed downsides.
Okay, and the last question, what about use of exogenous ketones?
I think they have the potential to help, but I do not look at them as being equivalent in terms of their therapeutic value as those ketones which are produced endogenously. The beta hydroxy ketone, that's really, which is produced endogenously, that's the gold standard, and I think that's what we really want to try to increase. That being said, there is some research to support the use of things like medium chain triglycerides or coconut oil for an exogenous source of ketones, and I think there's some research that would suggest it may have additional therapeutic value, but I wouldn't put everything in on that. I would try to get as many of the naturally or endogenously produced ketones from a higher fat, carbohydrate restricted, protein restricted diet, as opposed to just throwing medium chain triglycerides on top of a otherwise modest carbohydrate, modest fat, modest protein, typical macronutrient represented diet with extra medium chain triglycerides will not be as effective as a ketogenic diet.
Okay, thank you, John. And for all the questions that were not answered, we can put together a fact sheet with those questions and to be shared later on our website and through social media. So please be sure to be on the lookout for that. I'll send a follow-up email with those as well.
Thanks, everyone. Take care.